Hello. Sorry for the inconvenience, but yeah, I had some issues with my computer. Now I'm really happy to present the next speaker of this session, and I would like to present Professor Taube from the University of Fribourg. Who will talk about physical exercise and brain health. Please, the stage is yours.
Thank you for the kind introduction. Yeah, as said, I will speak about physical exercise and brain health with a special focus on the GABAergic of the inhibitory system. And what do we know so far about physical exercise and brain health? And I think we know just some things already. So, for example, we have some acute effects. Neurotrophic factors are released like BDNF, EGF1, VEGF. Then we have some neurotransmitters which are released like dopamine, serotonin. We have increased lactate, which is probably playing an important role, but it's not so clear yet. And we will decrease inflammatory markers. For the more long term, these substances will increase neuroplasticity. Neuroprotection will benefit from then. We have an increased neurogenesis, and our brain metabolism and vascularization is increased by physical exercise. And many of these aspects are mostly investigated in animals, or better investigated in animals, but on the behavioral level, we know that we have a good indication that cognition and learning will improve by physical exercise, that also motor control and motor learning benefits from it, and that the risk of neurodegenerative diseases is reduced. What we don't know so far specifically is what kind of exercise is doing what. So to specifically use one sport to promote a specific aspect of brain health, and I will come back to this later. Here is another important aspect. We've heard a lot about epigenetics today, and there is an example, physical exercise, how it promotes the transition from an inactive chromatin to an open state chromatin, so that in this case, the gene for BDNF can be expressed. So this is a transduction cascade we don't have to know, but in the end, we can say that with physical exercise, we can activate some genes which are important for our cognitive development. And in this case here, I think it's important for the parents to know if they do sports, it's positive for their offspring. But we also have a strong indication that this kind of training, this high intensity interval training, is promoting the expression of BDNF. So here, this link is pretty solid so far. Today, I want to speak about something which is not, you cannot read much about that. It's a mismatch between excitation and inhibition. And there are a lot of populations that face this risk. For example, with aging, we lose the ability of the inhibitory system. I don't know, do you hear the sound as well? I always have this sound in my ear. With aging, the inhibitory system is declining, sleeping disorders and insomnia. We have a decreased inhibition, also associated with developmental problems, some sorts of anxiety and depression, and some sorts of chronic pain. I come later to this. Many motor dysfunctions like dystonia or tics are associated with a mismatch between excitation and inhibition, and also coordinative skills like balance. So the question is, can we put this into balance to get a better brain health? And this will be the topic of my talk today. And first of all, what is intracortical inhibition? How is it mediated? So here you can see the pyramidal cell. If we excite that, we will have an excitation at the motor neuron and then the muscle. But we also have a huge network of inhibitory interneurons, and many of them, most of them, use GABA as the main neurotransmitter. It's thought that 1/3 of our neurons utilize this GABA. We have different methods of measuring it. We could use magnetic resonance spectroscopy. We can use EEG, or we can use paired pulse paradigms with transcranial magnetic stimulation. And due to time constraints, I will focus only on the last point here today. So how can we measure it? Here you can see a coil, and we can apply a pulse over the, in this case, motor leg area of the brain. So we can activate the neurons which are responsible for these muscles, and we can elicit a so-called motor evoked potential, MEP. And now we can influence this MEP by a conditioning pulse. This is a very small and subthreshold pulse. You can see it displayed here. This will not activate excitatory neurons. It will just activate the interneural network, the inhibitory network. And what you can see here is that we have a mismatch between these two, and the ratio between these two amplitudes, it's called SICI, or short interval intracortical inhibition. It displays the level of the intracortical inhibition. So now the big question is, can we train that? We've seen some populations with a deficit in intracortical inhibition. So here's a training study where we can see that it's muscle-specific, task-specific, but most importantly for us, also training-specific. In this study, we compared explosive strength training for 4 weeks with balance training on different devices, on different unstable devices. And you can see the outcomes here. So when we measure intracortical inhibition during activity, we can see that after the strength training, we have actually a decrease in inhibition. So there's a significant decrease, less inhibition. Whereas after the balance training, we have a significant increase in intracortical inhibition. This is summarized here. So if we measure during activity, we can see that intracortical inhibition is reduced after strength training, probably to maximize neural drive. But on the other hand, after balance training, we have an increase in intracortical inhibition, probably to better coordinate intra and intermuscular activity. Importantly, we don't see any effects when measuring at rest. So can we apply that now to these populations with deficits in intracortical inhibition? And my first example concerns aging. So if we look at the elderly population, we did a training study with them. So the intervention group, they trained for 6 months. After 2 months, they were measured. After 6 months, and after another 6 months of detraining, this means no training at all, they were measured again. They were compared to a control group that were measured at the same time points, but without following the intervention. We measured their balance abilities, so their postural control, and we measured also the intracortical inhibition, the amount of intracortical inhibition. What we can see here is balance performance, and shortly displayed, you can see that the balance training group reduced postural sway, so they improved balance performance, whereas the control group remained at the same level. And we also can see that there are significant differences between the two groups at any time points except baseline. Now the question which is more important for us, intracortical inhibition, what can we see here? And here we have a very nice finding that the intracortical inhibition increased in elderly people. That's the very first intervention where intracortical inhibition could be increased at all. So there's no intervention before could increase that consistently. And at the same time, the control group did not show this modulation. So they remained stable. Again, significant differences between groups. And I said before, the elderly people, they have a deficit in their intracortical inhibition, and that can be seen here. You can see here the level of intracortical inhibition in the elderly before the training, and here the level of young participants. So we measured also 40 young participants. And you can see that with training, the elderly subjects approached the young ones. My second example, how we can restore cortical inhibition, is in a pain patient. Due to Corona, we couldn't do a cohort study. We started with a single case study, but a very nice one, I think. 62-year-old patients with 4 years of chronic pain of unknown origin, and he didn't respond to any usual analgesic drugs, which was very bad for him. And then we did a study where we measured intracortical inhibition in different ways with TMS, the sticky parameters, and also with EEG, beta frequency bands. And we measured pain, behavior, and the GABAergic response after gabapentin, so after medication and after balance training. You can see already the results here. For the patient, the most important result was the pain. Here are different questionnaires for pain. And what you can see that gabapentin didn't do a large effect. So some were slightly improved, others did not respond to this gabapentin. Whereas the balance training did reduce pain. Then he did for several months detraining. He didn't do any training and everything worsened. And afterwards he did a second balance training and you can see that he improved again. And now the question which was more important for us, this was the intracortical inhibition. So we measured it during different situations. This was the baseline. After gabapentin, it was slightly increased, but then we had the greatest increase after the first balance training intervention. Then the detraining, it went down. And after the second boot of balance training, we had an increase in intracortical inhibition again. And very importantly for the patient, also after the second boot of balance training, the depression, anxiety, and insomnia scores went down considerably. So my last example would be sleep. Very shortly, we have a big study going on at the moment measuring sleep behavior. What did we see in our big study before? That we did in a subsample, we recorded a sleep quality and we saw that people slept better. And at the same time, we know that during sleep, here's someone sleeping and we're measuring TMS, you can see that the intracortical inhibition is changing, it's modulated. Actually, it's increasing. It's a little bit, the display is not so good. So this is a larger intracortical inhibition. And if you wake up, you release inhibition. So our idea is that balance training will train the modulatory capacity of intracortical inhibition and that patients or subject can better modulate this intracortical inhibition in their sleep also. So this would be, or this is one of the main topics of my group, to investigate, to counteract the mismatch between excitation and inhibition. And we think that this would be a very targeted intervention for many brain health problems. This is my group. Thank you very much for your attention.
Okay. So thank you. I'm just subbing for Emanuele, who was apparently lost a connection. Again, he has a problem with his computer. But we will discuss your topic later, since we are a little bit over time, 20 seconds during the panel discussion. And I apologize for the bells, but I think there was someone in the chat that I tried to address, but they were quite had some resilience. Okay. Thank you so much, and we'll be back. Bye bye.
See you. Bye.